In this study, we have investigated the neurobiological substrate of an important behavioral disorder that is food addiction. So, in this study, we asked why some individuals become food addict, whereas others are resilient to develop this behavioral disorder. So the strategy, the experimental strategy that you use is a translational approach. We have used in humans the IFAS-2 score to identify the behavioral characteristic of humans, and in animals, in rodents, we have developed a new behavioral model in our laboratory to identify food addiction in mice by mimicking exactly the same characteristics than food addiction in humans. So, with these approaches, we have identified extreme subpopulations of human and mice that are addicts or resilient to develop food addiction. And in this subpopulation, we have identified all the behavioral characteristics, and also we have investigated the microRNAs, because microRNAs are epigenetic factors that can represent a link between the genes and the environmental factors, because they are going to modify the expression of the gene depending on multiple factors, including the environmental factors, without modifying the sequence of the gene. So another important interest of the microRNAs is that in animals we can evaluate the microRNAs in the brain area that we want, but in humans we have also circulating microRNAs in the plasma. So we can make a translation between the miRNAs in mice in the different brain areas and the circulating miRNAs in humans.
So our objective was to investigate why some individuals are vulnerable and develop food addiction while other individuals are resilient and do not. To answer this question, we use a translational approach in this multifactorial complex disease. So in the animal study, we separate mice into two different subpopulations, addicted and non-addicted, and we found differential microRNAs that were differentially expressed in the medial prefrontal cortex, in the prelimbic area of this prefrontal cortex, specifically 29C and 665. Then, in the study in humans, we found that these two microRNAs were also differentially expressed in a subpopulation of addicted individuals. So this translational approach was accomplished by comparing these two different studies. So then the next step was to do a functional validation to demonstrate that these microRNAs had an important role in the development of food addiction. So then we performed another study in mice. We use a viral vector approach where we inject in the prelimbic medial prefrontal cortex virus that were inhibitor of microRNAs, specifically a tag decoy strategy that consists in inhibiting specific microRNAs. So we downregulated microRNA 29C and in another batch microRNA 665. What we found? We found that this mouse with this manipulation developed a behavior that was addictive-like behavior. So we were able to produce one single modification in one specific microRNA and to change the phenotype and to see a phenotype related to food addiction, specifically a phenotype of persistence to response in the case of the microRNA 29C, and also a specific phenotype of increased compulsive-like behavior in the case of microRNA 665.
The identification of this crucial role of this particular microRNA may provide novel biomarkers for an early diagnosis of the disease. And also we must remember that now we have also tools to modify the expression of messenger RNA. So in the future, this could also represent an important novel therapeutic strategy for this behavioral disorder.